And we were talking about selection on continuous traits. And I've already spoken about the three basic ways that can happen. I mean, you can have more complicated situations than this, but we talked about directional selection, where you're shifting the mean, stabilizing selection, where you're selecting against the extremes and favoring the mean, and disruptive selection, where you favor the extremes and select against the mean. And to go farther than that, I introduced the concept of variance, and I'm not actually going to make you calculate this on the exam, but you need to at least conceptually see how this works. The variance in a quantitative trait is the mean of the squared differences between each data point and the mean. The variance is also the square of the standard deviation, or if you want, the standard deviation is the square root of the variance. And intuitively, you can think of the variance as a measure of the width of the distribution. So if we had a bunch of students that averaged 5 feet 8, and they were all between 5'7" and 5'9", that would be an extremely low variance. On the other hand, if we had a bunch of people here and the average was 5'8", and the shortest of them was 4 feet 10, and the tallest of them was 7 feet 2, that would be a much wider variance, even if the mean was the same. And we can abbreviate the total variance by VP. Some of that variance comes from genetic variation in the population, and some of it comes from the effects of environment. So if we had a room full of people that ranged from 4 feet 10 to 7 feet 2, some of what makes those people different heights, some of the cause of why we've got such a wide range of people, is that people have inherited different genes. Short people tend to have short kids, and tall people tend to have tall kids. Not absolutely always, but it's a fairly strong tendency. Some of that variance comes from environmental variation. Some of the reasons, one of the things that can make people short is food restriction in childhood. One recent example, at the end of World War II, the Nazis had occupied the Netherlands, and there was hard fighting to drive them out. And one of the unfortunate consequences was what the Dutch still call the hunger winter. There was an entire winter season where there just wasn't any food at all, and people were trying to trap songbirds and eating their household pets, and a lot of people died. The kids who were children during the hunger winter reached adult heights that were measurably shorter than the average had been before. If you have an inconsistent food supply in childhood, one of the ways the body tries to adapt is you end up with a shorter height than you would have had if you had all the nutrition you wanted and to spare. So some of the reason for that spread between 4'10" and 7'2" people is down to environment. Some of that spread is due to genetics. And you can think of the total phenotypic variance as just the sum of genetic variance, or V sub G, and environmental variance, V sub E. Note, by the way, this is entirely relative to what population you're measuring it in. If I got a sample of students who were all college students from a middle-class American background, V sub E would probably be pretty low, because for all of the issues that the United States faces, episodic starvation really isn't one of them. If anything, if anything, people get too much rather than too little. And in a population like that, it's very likely that childhood starvation has not been a major factor in determining people's height. I mean, it could. There's always people with interesting backstories. But for a population like that, V sub E would probably be quite low. On the other hand, if I took a population from many countries, including some that were affluent and some where famine was still a real threat, in that population, V sub E would be greater, right? Dealing with a population that's coming from a more varied environmental background than if I were dealing with a population where everybody came from Little Rock's Heights district. To name a place just off the top of my head where childhood starvation is probably not a problem. Okay, so just remember that variance depends on the population that you measure it in. There's no table where you can look up and say, the variance VG for human height is always 0.8, right? Because the measure of it depends on the background of the people you're measuring it on. And you can think of, if you'd rather think of this graphically, V sub G would be the variance if all members of a population had an identical environment. You'll see this again. We do this a lot in what are called common garden experiments. And they're called that even if we're not using garden plants. But we could take a bunch of different tomato plants, let's say, and grow them all in exactly the same environment, in the same garden, same soil, same amount of water and fertilizer. Give each one an identical environment as much as we can. And variance due to environment would go to 0, or as close to 0 as we could get it if there's no difference in the environment that those plants are experiencing. And then all of our variance would be V sub G. On the other hand, we could take cuttings of one single tomato plant, which are genetically identical to each other, and grow them in a wide range of environments. And in that case, V sub G would be 0 because our plants are all clones of each other. There's no genetic variation at all. And the only variance that's left would be variance due to the environment. If we put our cuttings in different greenhouses with different amounts of water and sun and what have you. And then in a real world population, the total variance would be the sum of genetic and environmental variation, because in the real world you have both. Imagine going to the nature reserve and looking at the oak trees. They're genetically different from each other. They're also environmentally different because some might be getting a little more shade and some get more sun. And some are growing close to these little water-soaked patches that we have where the soil's a bit wetter. And some are in drier areas, and some have competition and some don't. So if we did a study like this on actual plants in the nature reserve, we'd have both variance due to genes and variance due to environment. And VP would be the sum of both. Okay, again, these apply only to variance within a population, and they don't apply to individual traits at all. You'll sometimes hear people say that 70% of your height is due to genes. That's meaningless. 100% of your height is due to genes and to the environment, because you can't disentangle them that way. If I'm 6 feet tall, there is no meaningful way that I can say, well, I got about 5 feet of my height from genetics and the other foot from eating weeds. That's meaningless. You can't apply this to an individual. There's no way you can say my IQ is such and such, and 80% of that comes from the fact that I've got smart parents, and 20% of that comes from the fact that they enrolled me in Montessori school. Okay, it doesn't work that way. You're only talking about variance in a population. It's like fitness. It's not the kind of thing that an individual has. You're looking at a statistical measure based on a population of individuals. It's not an individual property at all. And again, they also don't apply to variance between different populations. There's no, we could calculate VG for height in the population of UCA, but we could do the same thing in a population of, I don't know, unfortunate refugees in Somalia, and we might get a completely different answer. These measurements are always, always, always relative to the specific population that's under study. And then we can take VG divided by VP, the fraction of variance in a population that's down to G's, and that quotient is called broad sense heritability, abbreviated capital H squared. And you have to be careful with that because a trait can be inherited genetically and have zero heritability. A trait can even be fully genetically controlled and have undefinable heritability. Let me do that one first. Everybody in this room has one nose. Yes, right? Nobody's got any extras anywhere? Nobody brought an extra nose to school today? Okay, we could measure the number of noses per person. 1, 1, 1, 1, 1, 1. All right. So that is a total of 6 noses divided by 6 people. So the average number of noses is 1. The total variance in the population, we take each trait, each measurement, and take the difference between each measurement and the mean, and then square those differences and add them up. So you have 1 nose, the average is 1 nose, so the difference between your number of noses and the average number of noses is 0. Your difference between your number of noses and the average is 0. 0, 0, 0, 0, right? So the variance in the trait is 0. Yes? So if the VP for number of noses is 0, VG for number of noses is also 0. There's no genetic variance in the number of noses. So our broad sense heritability is 0 divided by 0, right? Which is undefined. It doesn't exist. We know that having a nose is very much under genetic control. There's a number of birth defects that can leave you without a nose or with a badly misshapen one or extremely small one or something like that. We've identified some of the genes that are involved in shaping the developing face. So we know that having a nose has a genetic background, but we can't define its heritability because there's no variance in the trait. Okay, am I making sense? So when we say that such and such is genetic, when people say that, if people say, yeah, well, your intelligence is genetic, or your height's genetic, or something like that, what they usually, they're getting something wrong. You can say that these traits have a high heritability in a certain population. That's not the same thing as saying that they're controlled by your genes. Because things like the number of your noses is controlled by genes, and yet it doesn't have a heritability we could measure. And a trait could be inherited genetically and have 0 heritability if it's got no genetic variance. So imagine we have a population of cloned people who all are born with exactly the same skin color and all have exactly the same alleles for the at least 9 genes that are important for determining the color of your skin. In that case, VG would be 0. However, We could still have phenotypic variance if some of those people stay away from the light, like they're goths or something, and some of those people are hitting the tanning salon all the time. Or if they're lucky, hitting the beach all the time. That causes variance in skin color, right? If some people go out and get a savage tan and some people don't. But none of that variance would be due to genes. And yet, we do know that genes do affect skin color. But remember that that's always relative to the population that you're studying. If we're studying a population of people who come from all over the world, then we would find genetic variance in skin color. But if we're studying a population where everybody has identical genes and yet some of them tan and some of them stay out of the light, within that population where everybody's genetically the same, we'd have zero heritability. Okay, am I sort of making sense? All right. Here's where people sometimes fall off the rails a little bit. There is, okay, in when you take genetic variability, you can break that down into 3 different components. Variation due to genes, you can subdivide into additive genetic variance, dominance genetic variance, and interaction genetic variance. What the heck did I just say? All right. Additive genetic variance is genetic variance that's explained purely by the copy number of genes that you've got. Okay, quick example. In kitty cats, somebody, she may not be here, but I was having this conversation yesterday. In domestic cats, white spotting is governed by a single gene with 2 alleles. And spotting is dominant to non-spotting. If you are a kitty cat with the genotype, if you've got 2 copies of the no spotting allele, little less, then you don't have white spots. You are a solid color, whatever you are, black, orange, brown, whatever. If you have 2 copies of the spotting allele, you have so many white spots that you might be more white than anything else. A cat that's homozygous dominant for the spotting allele is going to have mostly white on its body and maybe just the top of its head and its ears with color. Or maybe the top of the head, its ears, and a little bit of its back having color. A heterozygous cat is going to be mostly solid color and might just have white feet, or maybe a little white star, or like one of our cats, it might be completely black, except it's got 1 little white spot on its belly. We call it the reset button. Or might have the tuxedo pattern where you've got, some white on the face and then kind of a white diamond on the chest, and white feet makes the cat look like he's wearing a tux. All right. So the amount of white spotting on a cat is entirely additive genetic variance because it entirely depends on how many copies of the spotting allele the cat's got. If the cat has 0 copies of the spotting allele, it doesn't have any white on it. If it's got 1 copy of the spotting allele, it has a little white on it. If it's got 2 copies of the spotting allele, it has a lot of white on it. Am I making sense there? So in that particular case, the variance in the trait is due entirely to additive genetic variance. It's due only to the number of alleles that an individual cat has. Dominance genetic variance is variance in a trait that comes from the fact that some of these alleles are dominant and completely cover others up. And then interaction genetic variance comes from epistasis. What? genes interacting with each other. What's a good kitty cat example? Okay, here we go. A good example from cats is you've got one gene with 3 alleles that determine whether your cat's basic color is black, dark brown, or light brown. With me there so far? There's a second gene that will lighten whatever the base color is. It's called the dilute gene. So a cat that's got 2 copies of the black allele, but also 2 copies of the dilute allele is going to look gray. There's no gene for gray coat color in cats. There's a gene for black and a gene for lighten this color up. And a cat that has 2 copies of the allele for light brown fur and also has the gene for dilute is going to have this very, very faint brown color that they call, I think breeders call it lilac for some reason. So in that case, when you're looking at cat colors, you can't look at single genes. You have to look at the way that genes interact with each other. So if we were doing an experiment on cat color, and we had a whole bunch of cats, and we came up with some way to measure what color they are, some of what makes those cats have different color patterns is due to the additive effects of genes. Some of it is due to dominance effects of genes, and some of it is due to epistasis, the interaction effects of genes. All right, here's another example, yes?
What about those cats that have like color inside them?
Oh, okay, like a calico?
No, like
Oh, interesting.
full and have that.
Okay, like two-faced cats?
Basically, yeah.
Okay. If you have a cat that's like black on one half the body and white on the other half, it is possible that when that cat was still 2 cells, one of the cells underwent a mutation and the other one didn't. It's possible that the cat could be a chimera. Sometimes you will get 2 embryos that fuse in development and you end up with cells from both. This actually happened to a friend of mine. She didn't find out until she was middle-aged that even though she identified as female, some of her cells were XY. She had started out as a pair of twins, but at a very early age, the embryos had fused together, and she was actually a genetic mixture. That may happen more often than we think. So kind of, she had some adjustment to go through because socially she's female. Anatomically, I have no idea. We're not that kind of friend, and I'm not that interested in finding out. But she thought of herself as female as far as I know. And yet here she is, she's got some of the cells in her body are actually XY. Which means in North Carolina, she can't go to the bathroom anywhere. But yeah, this kind of thing probably happens more often than we think because it's not the kind of thing that you get tested for at every physical. It's not the sort of test the doctor's going to order unless the doctor feels there's a specific reason for it.
How did she find out?
What's that?
How did she find out?
Off the bat, I don't recall. The doctor might have got. There may have been something that made the doctor suspicious. I'm not sure what. And again, we're friends, but I'm not that familiar with her, so it's not really the kind of thing I asked about. She's a cool person, and that's all I really need to know. Right. Yeah, this is okay. Look up. What the heck? I will. Let me go to you look up gynandromorph cardinal, yeah, you can occasionally get a female egg cell that accidentally develops with 2 nuclei, and one of them gets fertilized, and they get, and this is why this is really rare. You end up with an egg cell that's accidentally got 2 nuclei, and the egg also gets fertilized by 2 sperm at the same time. Normally that's not supposed to happen, but if it does, you can end up with an individual that is quite literally half and half. So that's a cardinal that was found in Pennsylvania not too long ago, where half of the bird's body is male and the other half is female. And again, that 2 unlikely things have to happen at the same time in order to get something like this. You have to have an egg that's accidentally got 2 nuclei because something in meiosis was off. And you have to get double fertilization, which is also not supposed to happen because egg cells have mechanisms that are supposed to keep that from happening. And yeah, basically, 2 unlikely things have to happen, which is why you don't see this very often. But when it does, you get half and half. I know it's been documented in cardinals. I know it shows up in fruit flies. It's probably happened in other cases. Don't know if it has ever happened in humans, although it wouldn't really surprise me. Humans can develop in pretty interesting ways sometimes. But yeah, it is also conceivable that that cat you showed me is a gynandromorph. But again, I don't know. All right. Okay. Let's go back to slides. And let's do a quick, I'm going to pull up the whiteboard in just a second. This is an insect called the scarlet tiger moth. It's European, I believe. And it's got spots on the forewing. And the number of spots is governed by a single gene with 2 incompletely dominant alleles. So the bottom moth has 2 copies of the recessive allele, and it's only got 2 large white spots per wing, right? You see them? Most of the wing is black. There's a few tiny little flecks, but each wing only has 2 big spots. The one in the middle that is heterozygous, each front wing has 4 big spots. And the one at the top, homozygous dominant, for my purposes, I'm going to count those moths up at the top as having 6 white spots per wing. Maybe a couple of the spots are kind of fused together, but I'm doing the counting. I've got the PhD. Those moths have 6 spots per wing. Okay. What I want to show you is how we can measure additive genetic variance. And it's not that hard if you can draw graphs. So let's cut over to sharing the whiteboard. All right, so just as a reminder, we have these moths, and if they have genotype big S, we have 6 spots. Genotype big S, little s moths have 4 spots on each front wing, and little s moths have 2 spots per wing. All right, what we can now do is draw a graph, and we're going to envision different crosses we could make between moths. And we're going to graph on the x-axis. We're going to graph on the, on the x-axis. The mean parental phenotype. And of course, I don't mean the phenotype of mean parents. I mean the mean phenotype of the parents. And on the y-axis, we're going to put the mean offspring phenotype. And let's put some numbers here. Put them in a different color. Why not? Maybe a different, do this in a different font. 2, 4, 6. And here we'll do the same. We'll graph. Okay, close enough. Okay, so now start thinking of all the possible crosses you could make. Let's say we cross big S with big S. Okay, if we cross big S, what's the average number of spots for each parent? It's 6, right? Okay. How many spots are the offspring going to have? They're all going to have 6. So we can put a point right there. Okay. What if we cross 2 little s parents? What's the mean parental phenotype? It's 2. All of the offspring are going to look like how? All of the offspring will have 2 spots. So we can put another point right there. All right, let's say we cross a big S, and a little s moth. What's the mean parental phenotype? 4. If we cross 1 with 6 and 1 with 2, the average of 6 and 2 is 4. Yes. Is that a question or you're signaling the number 4? Yeah. Okay. All right, if we cross a big S, and a little s, what will all the offspring have? Yeah, they will all be big S, and they will all have 4 spots. So we can put another spot right there. Okay, watch this. Suppose we cross big S with big S. So one parent has 6 and one parent has 4. What do we get? The average is 5. All right. If we cross a big S with a big S, half the offspring will have 6 and half will have 4. What's their average? 5. All right. So we can put a point right there. If we cross big S with little s, the parent's mean phenotype is 3, and the offspring's mean phenotype is going to be 3. If we cross 2 that have 4 spots, we're going to end up with 1 with 6, 2 with 4, and 1 with 2 on average. So take the average of 6, 4, 4, and 2. That's 16 divided by 4, which is also 4. So we can put another spot right. We can put another spot right there on top of that one. For every cross you can make, the mean parental phenotype and the mean offspring phenotype are always going to fall along this line. Okay, I didn't draw it perfectly straight, but right, you can see it. And the line, of course, is fits the equation y equals x. And then, of course, the slope of the line is equal to exactly 1. Okay, are you with me there so far? Everybody see how I did that? The slope of that line is the measurement of additive genetic variance. Okay, all of the variance here in the setup that we've given, we assume it's all genetic, although it might be interesting to raise tiger moths in different environments and see if they all spot the same way. But assuming all the variance is genetic, it is all additive genetic variance, because the only thing that governs how many spots a moth has is the number of big S alleles that it's got. Okay, you with me there? You see where I'm getting at? That is genetic variance that is completely additive. Now, suppose for a minute we were looking at a different species. And let us say that in this different species, we had complete dominance. So if you are homozygous dominant or heterozygous, you end up with 6 spots. What does our graph look like now? Well, let's draw it. Okay, if we cross a 2 little s moths, we get little s offspring. So there's a point there where the axis is 2 and the y is also 2. It's like I accidentally erased those letters on the y-axis, but just imagine they're there. If I cross 2 big S moths, then I get all big S offspring. So that's a point there, right? If I cross a big S and a little s, though, the mean parental phenotype is 4, but all of the offspring will be heterozygous and they'll all have 6. So I end up with a point where the mean parental phenotype's 4, the offspring have 6. So that point's up there. If I cross big S with little s, the mean parental phenotype is 4, right? The average of 6 and 2. But if I cross big S with big S with little s, the mean parental is 4, but half the offspring will have 6 and half the offspring will have 2. So we have a point right there. Let me see, if I cross big S, okay, if I cross 2 big S, little s together, the mean parental phenotype is 6, but we're going to end up with 3/4 of the offspring with 6 and 1/4 of the offspring with 2. So that's going to be 20 divided by 4, which is actually going to be 5. So we end up with a spot right there. What I hope you're seeing is that we're not getting all these points lying on one line, right? We're getting scatter. And that scatter is due to the fact that in this species of moth, we have to take dominance into account. And in fact, if we were to draw the best fit line, we would end up with something like this. I think the slope of the line might be, I've seen this done in the real world, the slope comes out to something like 0.8. I don't remember exactly what it is, but the slope is no longer 1. So some of what's making our moths different from each other is due to additive variance. because it's got to do with the number of alleles that you've got. But some of what's making our moths different is due to genetic dominance. The fact that in this species, spotting is completely dominant over less spotting. Okay, so the way that we can measure additive genetic variance is by doing a graph of mean parental versus mean offspring phenotype and calculating the slope of the best fit line. All right, and in fact, we're going to do this in, yes?
Oh, sorry. Oh,
Okay, we're going to do this in class because tomorrow I want you to bring me some data. Monday, I want you to bring me some data. Okay? All right. If you can, there may be some students who have family situations where they can't gather this data. That is perfectly okay. Nobody will be penalized if you don't have it. But if you're able to, bring me on Monday your height. I'll convert it to inches because that's the way I've been doing this for the past 10 years. If what you have is centimeters, I can, we can work with that. The height of all of your full blood siblings, if you have any, and the heights of your mother and your father. Again, if you're adopted or you have relatives that you don't know how tall they are, or you have kin that you're not speaking to and you can't call up and ask, hey, how tall are you? That's cool. You don't even have to tell me. Different people have different situations, and I'm not going to store any identifying information with this. But for the past at least 10 years, I've been gathering data from every time I teach this evolution class. And we're going to estimate the additive genetic variance on human height among a population of UCA biology majors. Okie doke? Yes.
so for VA, the additive genetic variance, for the last one would be what? This one would be 0.8-ish.
Yeah, for the first example that I worked, the additive genetic variance was 1. The only thing that was making those moths have different spot numbers is the number of big S alleles that they have. And then in the second example, I changed it. I don't know if this corresponds to a real species, but I changed it to a species that has complete dominance of the spotted allele. And in that case, we get an estimate of additive genetic variance as less than 1. I think it does come to 0.8. I don't remember exactly what it would be. And I don't have spreadsheet functions on this whiteboard. I could work it out. I just input some numbers into Excel and make it fit the line. But yeah, our slope of the best fit least squares linear regression, our slope of the best fitting line is our estimate of additive genetic variance. And here you can see it is less than 1. So some of what makes these moths look different from each other is due to the additive effects of genes. Some of it is due to the fact that in this case, we have complete dominance of one allele over the other allele. And then things would get weird if we had interactive genetic variance. If we had one set of genes for moth wing spots and another set that influenced how the first gene worked in some way. I'm not even going to try to get into that. I'm not a moth spotologist, but it could get more complicated than this. But yeah, the slope of the best fitting line is going to be your estimate of additive genetic variance. Let's go mercifully back to the slides for just a sec. Yeah, graph mean parental against mean offspring phenotype for every possible cross, and you'll get a slope of 1. That means all of the variance in wing spotting in these moths is additive genetic variance. So the fraction of genetic variance that's additive, V sub A, divided by the total population phenotype variance, that V sub P that we started out with in the beginning, we write as the narrow sense heritability. I might actually have dropped out a slide here. But yeah, if we take that narrow sense, okay. I did? Let's see, okay. All right, V sub A divided by V sub P. The additive genetic variability divided by the total variance is something called narrow sense heritability, and we abbreviate that as lowercase h squared. Why the hell would we ever want to do this? I can hear you asking yourselves that question even now.
Why the hell would we even want to do this?
I'm glad you asked. Why the hell would we ever want to do this? The reason we want to do this is that we can plug this into something called the breeder's equation. And this is the kind of thing that is very much practically used in animal husbandry. This is the kind of thing that animal and plant breeders take into account. When I was little, I spent a lot of time on the Mississippi State University campus because my grandfather taught there. And don't laugh, they've got a big time program in poultry science. And I'm sure they teach this in the poultry science classes quite a lot. What that narrow sense heritability is good for is predicting whether or not selection is going to be effective in changing the mean value of a trait in a population. So we can define it like this. Let's say we've got a population of, what the heck, chickens. All right. And we measure, what do we want to breed our chickens for? Do you like eggs or meat better? Eggs. Okay. We want to breed our chickens for better egg production. In fact, I tell you what, you can finish copying this down later. Let's go to the whiteboard. And let's erase all that. All right, we're working with chickens now, and we have some measurements of egg production. Let's say it's, I don't know, number of eggs. Do that in a brighter color. Dark green would be lovely. Okay, number of eggs per month. And on the y-axis, we'll put number of chickens. All right, so we've got our flock. We've got lots of chickens. And for each chicken, we're measuring the number of eggs that they lay per month. And this is a trait, and it's got variance. And let's assume that the variance is what we call normally distributed, meaning that it fits a bell curve. So let's say that some of our chickens are real slackers and might only be laying a couple of eggs a month. And most of our chickens are laying kind of a medium number of eggs per month. And then a few of our chickens are just cranking the things out on some kind of ovarian assembly line. Okay, so we have a few chickens that are bad layers, and we have a few chickens that are champion egg layers, and most chickens are kind of there in the middle. Okay, you with me there so far? All right, yes.
What are the axes being switched?
Sorry?
What are the axes being swapped and chickens are being? Right, because I'm sorry.
Okay.
I'm having trouble visualizing this.
Okay. we have like one chicken that lays, this probably would actually work a little better if I'd drawn it as a bar graph. We have one chicken that's laying 2 eggs per month and 3 chickens that are laying, 6 eggs per month. And we have 50 chickens that are laying 30 eggs per month. And we have one chicken that's laying 60 eggs per month. Okay, so we're not measuring the number of eggs per chicken. We're measuring the number of chickens that are laying particular numbers of eggs. Okay, so our good layers are over here to the right, and our poor layers are over here on the left. And being good statistician, dang it. Really ought to be there. There's undo. Okay, that'll have to do. Okay, being good statisticians, we can calculate for that population the average number of eggs per month. And it is, let's just call the number of eggs per month X. And the number of eggs laid per month, we can just write X bar, right? Remember, a bar over a letter means it's an average. All right, with me there. Now let's say we decide, okay, all of our chickens that are not laying eggs are going to end up in chicken fricassee, or whatever it is you do with chickens that you don't want. And we're going to save only the top layers. So we decide that right here is a cutoff. If you're a chicken and you're not above the cutoff, then you get cut off. Get it? All right. So if you are a chicken and you're laying fewer than this number of eggs per month, you end up in stew. If you're laying more than this number of eggs per month, you get to live and you get to breed. All right, with me there. I'm going to go ahead and shade that part. Now, what the heck? I'll shade that part green. Okay, those are the chickens that are allowed to survive and keep breeding. The rest end up in the stew. There we go. And after we have gotten rid of all of our inefficient chickens, we can calculate the mean number of eggs laid per month for the survivors after selection. And we can call that X bar star. Sorry, that's not looking very good. That's supposed to be an asterisk. Okay, you with me there? All right. The selection differential, I'll even put this in somewhat bigger letters. Equals the absolute value of X bar minus X bar star. All right. So the selection differential is equal to the absolute value of the average trait before, the average of the trait before you do selection and the average of the trait after you do selection. Think about it. If you said, I want most of my chickens to live and only a few of the worst are going to get end up as chicken stew, then your cutoff value might be here. And the mean value after you've gotten rid of these few chickens on the left might be very close to the mean value before you got rid of any chickens at all, right? On the other hand, if you said, let's try the Harvard University approach, only a handful of the overachievers are ever going to get in, and to heck with everybody else. And you selected only like those chickens, only the 2 or 3 that were cranking out a stupid number of eggs per month, then there'd be a much greater difference between X bar and X bar star, right? So the selection differential is basically a measurement of how stringently you're carrying out selection in your flock. All right, you with me there? Okay, let's say you take the survivors and you let them breed, and you have, you end up with a new flock of chickens. Okay, so our new flock, I'm gonna do in, let's do it in red. So our new flock is in red, and you've gotten rid of your slacker chickens, and the ones that survived have bred you a new flock, and their number of eggs per month looks like that. Okay, with me there? It's directional selection. That whole bell curve has shifted, right? You've gotten rid of your slacker chickens, and they're not passing on their slacker egg genes. And you've encouraged to breed your, hardworking egg-laying chickens, and those have had lots of babies. And so you have a flock now that on average is producing more eggs, but there is still variance. You still get some chickens over here that are not really enthusiastic about it, and some chickens that are just, freaking, Harvard hopefuls over here. They're not just laying eggs. They were like, president of the National Egg Honor Society when they were chicks, and they play the violin and solve world hunger and all that. They're the ones that get into the honors college. And then most of your new flock of chickens are kind of in the middle, just, raising, cranking out the eggs and, just hoping to save enough for retirement or whatever it is. So the mean of our new population, we can call Y bar. And the difference between X bar and Y bar, that difference right there, is a measurement of how effective your selection was. So R is the response to selection, and it's equal to the absolute value of Y bar minus X bar. And give me a second to draw the bars. Okay, so the selection differential is a measurement of how strong your selective pressure is. R is the response to selection. R is a measure of how strong the effects of your selection were. And the breeder's equation is equal to R equals narrow sense heritability times the selection differential. If narrow sense heritability is equal to 1, then if you apply very stringent selection, you're going to get a very large response to it. If the narrow sense heritability is 0, or if it's very low, then you can select as stringently as you want, and you're not going to get very much. You're not going to get much of a response. There's an old story. I'm willing to believe it's not true, but there was supposedly a turkey farmer in the 19th century who got the great idea of making all of his turkeys wear turbans. So he put little turbans on the top of each of his turkeys' heads. And the story is he believed that if he did that, his turkeys would lay eggs that would hatch into turkeys that were born with little turbans on their heads. Okay, The breeder's equation is why this does not work. The trait wearing a turban on your head has 0 additive genetic variance. In fact, it's got 0 heritability at all, right? There is no gene that will make you wear a turban on your head. You know, it is entirely up to whether you feel like putting on a turban or not. You know, that's a behavior that some people learn and some people don't, but it's got nothing to do with your genes. Anybody can wear a turban. So in that case, the narrow sense heritability of turban wearing in turkeys is 0. So it does not matter how strongly our turkey farmer tries to select only the turkeys that are wearing the finest turbans. It will do absolutely nothing because H squared is equal to 0. Okay, am I making a certain degree of sense there? I mentioned there've been, okay, this guy named Thomas Bouchard at University of Minnesota has been running a very extensive study for many years on identical twins reared in separate households. And I'm not going to go into how he does it, but you can use pedigree data from families like that as another way to estimate narrow sense heritability. It turns out that the specific religion that you are turns out to have essentially no narrow sense heritability. And that means if we tried to select the human population to raise only Baptists, it would have no effect at all. The religion that you are is about as genetically controlled as whether you wear a turban if you're a turkey. So that's another trait where selection simply wouldn't have any effect. The standard, I can't remember what the narrow sense heritability for egg laying in typical chickens is, but somebody's published it, and if memory serves, it's about 0.7. Meaning that if you did this with a flock of chickens, you'd get a pretty strong response. It might be worth your while to cull a lot of your chickens and only keep the really big egg layers. And because you get a large response, you get a lot of chickens that are laying more eggs than they would have if you hadn't done it. So the breeder's equation is what breeders will use to try to tell them if a selective breeding program is likely to have a useful effect. Okay, everybody got that? Okay, Ruby. All right, let me go back to slides. Actually, it's 10:18. Would you care for, that's actually a fairly good place to take a break. All right, would you care for a break? All right, what would you like broken? What's that? Yeah, shall we continue? Just trying to be very scientifically exact here. Even when I'm telling my yolks. Exactly. All right. I'm going to stop the recording.